Thank you so very much, and thank you for the kind words. Very appreciated. So, around 4.4 billion years ago, water appeared on Earth. And then, perhaps only a few 100 million years later, life formed in this warm and salty oceans of our little blue planet. And so it was for a few billion years more until life eventually climbed ashore and evolved into the forms of human beings, likely cognitively unique forms of beings that we humans are today. And ever since humans and the waters of the sea have had a special bond, the sea with its open oceans, bays, channels, and canals have brought inspiration and hope, adventure and danger for the heroes and nymphs of Greek mythology, for the spice route merchants of the Middle Ages, and for modern day transport. And in a way, this is a talk about the foundations of life. It is a talk about water. It's a talk about salt, about transport, and the interplay in the human brain. So our brains, your brain, my brain, roughly 80% water. It is surrounded by a narrow sea. On the outside is the so-called subarachnoid space. And deep inside lie the larger fluid-filled ventricles. And the brain pulsates. As your heart beats, blood flows up and into the cranium. The intracranial pressure increases, pushing the cerebrospinal fluid, which surrounds the brain, down into the spinal canal surrounding the spinal cord. And then venous blood starts flowing out of the brain and it pushes the cerebrospinal fluid back up. And as you breathe in, your thorax pressure increases and you have a similar pattern. And so your brain and its fluid-filled, its water environment wave back and forth in synchrony with both the cardiac and the respiratory cycle. These are, what you're looking at here are fluid structure simulations of this interplay in response to cardiac pulsatility. These are fluid structure poroelastic Stokes type simulations created by one of my PhD students, Marius Kausman. So, in fact, blood vessels are almost everywhere in the brain. They run along the surface of the brain, they dive into the brain, and they branch into a network of arteries, capillaries, and veins. In fact, any brain cell is likely less than 20 micrometers away from a blood vessel. But in spite of this proximity, the transport and exchange between the tissue and the blood is very tightly regulated due to the existence of the blood-brain barrier. And due to this barrier, it was traditionally believed that if you have a solute, meaning a drug or a dye or some kind of tracer in the cerebrospinal fluid or in the blood, that would not reach into the brain. That was the traditional belief, but this view is now changing. And it's changing in part because of what you see here. So these are so-called contrast-enhanced magnetic resonance, MR images, MRI, of tracer concentrations in human brains. There are two rows, those represent different views, so from the side and from the front. And the four columns represent different snapshots in time. So over a period of 24 hours. And this tracer has been injected in the spinal canal and it's traveled up, and as you can tell, around and into the brain over a course of 24 hours. And this tracer is a dye that's not able to cross the blood-brain barrier, but instead, as I mentioned, it's injected into the cerebrospinal fluid. And if you've seen the later images, you would also see that this tracer, this dye, is also cleared from the brain in the course of 24 to 48 hours. But if this tracer is not able to cross the blood-brain barrier, how is it able to get in, really? And how does it get out? Now, in the rest of your body, you have a lymphatic system. It consists of lymphatic vessels and nodes. And this system is named by the Roman goddess of water, Lympha, or the Greek equivalent, the water nymphs. This lymphatic system serves many roles, but one of its roles is to, very informally put, get stuff out of the tissue. But in the brain, there are, in fact, no lymph vessels. So the brain is, from a fluid mechanics point of view, characterized in part by a blood-brain barrier and a lack of lymphatic vessels. So it's almost as if there's no way in and no way out. Yet we still see, and you saw in the previous slide, that this tracer is transported in and it's transported out. And the big question here is how. And what I think is the intriguing answer is that no one really knows. And I think this is intriguing because it's a long-standing question, and it's a question that's literally very close to us. It's not as though this is a galaxy that's far, far away. It's really literally the transport mechanisms inside our own minds. But still, this question remains. So what will the rest of this talk will be about showcasing examples from our research on how we use computational mathematics to address this question. So let me start with how do solutes travel around and into the brain. So let's start by looking at some evidence, some videos. So what is this? So these are in vivo 2-photon imaging recordings, now in mice, from the leading group in the world in this area, the leading group of Maiken Nergård and her lab in Copenhagen and Rochester. And what they do is they inject this fluorescent tracer, again in the subarachnoid space, the fluid-filled space surrounding the brains of mice, and they observe how it moves over a time course now in minutes. This video shows 3 planes, one on the surface of the brain and then some hundreds of micrometers into the brain. It's in vivo, so it's a living, breathing mouse here. And what you can see is that this green tracer seems to first spread along the vasculature on the surface of the brain before it moves into the brain. And again, if you look very closely on the lower plots, you can see it seems to spread kind of along the vasculature, along what we call perivascular spaces. Okay, so let's look at this other video on the right here. This is in real time, and this also shows a mouse experiment, now with a bit larger particles, so-called microspheres, that you're able to track the motion of the individual particles. And here they've been able to track the particle trajectories and how the particle motion aligns with the cardiac and respiratory cycles. And looking at the spatial and temporal scale here, so this is some tens of micrometers, and this is essentially real time. So we see that these particles maybe move at the velocity of some tens of micrometers per second in these spaces. So that's the order of magnitude of the velocities we're talking about here. And so we can ask a very specific question, and it's a question that's been very popular in this field for the last 10 years. What are the driving forces? What are the mechanisms underlying this motion of particles, of tracers in the perivascular spaces? So conceptually, we are now looking at a space surrounding a network of blood vessels, and zooming in, for instance, in A here, we're looking at the perivascular space in B surrounding a blood vessel segment. The hypothesis is that these perivascular spaces are kind of either anatomically distinct or just kind of functional compartments surrounding the vasculature. And it's also believed to be then filled with CSF, cerebrospinal fluid, or interstitial fluid, ISF, for our purposes, water. And the dimensions and scales involved indicate that we're in a rather straightforward low Reynolds, low Wormsley number regime. So we can estimate the fluid velocity and pressure in this perivascular space, for instance, via numerical simulations, subject to different driving forces, including, for instance, the cardiac motion of the arterial wall, maybe other motions of the wall of the surrounding tissue, or other pulsatile or static pressure differences at the perivascular inlet or outlet. And one hypothesis has been very popular. It's a very natural, it's a very appealing hypothesis. And it states that could it be that it really is these rhythmic expansion and contractions of the arterial wall that induces the flow of cerebrospinal fluid in the perivascular space? Now that seems plausible, but in order for this to be an effective transport mechanism, it can't just be a pulsatile flow. There must be some kind of directional component to the flow. So the question becomes, can this arterial expansion and contraction induce a net flow in the perivascular spaces? So let's consider a thought experiment where we just consider an annular cylinder, so a cylinder within a cylinder, of length L, and we just consider Stokes flow in this domain. And it's driven by the motion of the inner wall by a wave with some frequency, and this wave travels down the length of your annular channel with a wave speed c. Now the mouse speed beats at around 10 hertz. The pulse wave is rather quick, around 1 meter per second or 1,000 millimeters per second, which gives a wavelength of about 100 millimeters. So now what type of flow will you expect in this channel if you have this, this type of wave characteristics for the motion? And I will say that this would strongly depend on the length of the channel. This is essentially what we've illustrated in plot B here, which shows time, position over time. So if we imagine that we suspend the particle in this channel and look at how it moves over time, this is what's shown in B for different channel lengths. So for long domains, we'll see large pulsatile velocities with a small but perhaps non-negligible net flow velocity component of some micrometers per second. But for short vessels, the wave motion is essentially simultaneous in the whole channel, and the pulsation will still induce pulsatile flow, but really no net flow. So here we see that we can get net flow if we are in channels that are half, 1 or 2 wavelengths of the wave. So here we're talking about something that's maybe 10 or 20 centimeters, but your mouse brain is maybe max 1 centimeter big, max 10 millimeter big. So we and many others therefore concluded that this cardiac-induced wall motion is not an effective driving mechanism for perivascular net flow, which still leaves the question of what is. So the simplest example would of course be, or the simplest explanation would of course be that there exist some kind of pressure gradients inside the brain and along the perivascular spaces. And clearly there is a pressure gradient between your arterial pressure inside the brain and the venous pressure, clearly, but it's not clear if there is a substantial pressure gradient within the tissue itself. And this is again a question that's been asked many times, and the traditional answer is that no, there's no pressure differences within the tissue itself. And that is a reasonable view, because if you implant a pressure sensor, like our clinical collaborators do in some patient groups, they implant actually 2 pressure sensors, one deep inside the ventricles of the brain, and one in either the subarachnoid space here or further into the tissue. And if you measure the pressure over time in these 2 locations, you'll get plots similar to B and C here, with a very characteristic curve. And the 2 curves in these 2 locations are virtually identical. And so there's consensus, and it seems unlikely that there are large intracranial pressure gradients, but we don't really need a very big gradient in order to drive the flow on the scales that we are discussing here. We just need some millimeters of mercury per meter, really. So what we've done is that we've very carefully analyzed a series of simultaneous recordings of the intracranial pressure, like the curves in B and C here. We very carefully analyzed a bunch of these curves for over 24 hours from these 2 brain locations in several individuals. And these are data, as I mentioned, from our clinical collaborators, Pekrist Eide and Geirings at Oslo University Hospital. And then we really looked carefully at the differences in this pressure. And what we found was that there was, in fact, a small but persistent pressure difference with two main frequencies. One at around 1 Hertz, corresponding to the heartbeat, and one at around 0.25 Hertz, corresponding to respiration. And we found that the peak amplitude of the cardiac cycle was, on average, around 1.5 millimeters of mercury per meter, or more precisely 1.46 millimeters of mercury per meter for the cardiac component, and around 0.5 millimeters of mercury for the respiratory component. So we were unable to measure static pressure differences because that was not accounted for, the positions of the patients and so on. But we were hoping that these pulsatile pressure differences that we measured could tell us something about the order of magnitude that can be expected. So in fact, what we see is that if we combine this arterial wall motion that we simulated first, together with a static pressure gradient of 1.46 millimeters of mercury per meter, we will in fact obtain traces of particles that are very comparable to what they observe experimentally. So in plot B here, showing position over time for different particles, here P1, P2, and P3 are different experimentally measured particles, while the inlet and outlet denote particles at the inlet and outlet of the PVS respectively. And in this case, we do observe rather similar behavior. And so this, I think, is our best answer at the moment, that perivascular solid transport is plausible, and it's driven by wall motion in combination with the net cerebrospinal fluid flow induced by something that yields a pressure gradient of the order of 1.5 millimeter mercury per meter. So now I've looked at particles moving towards and into the brain in perivascular spaces. But do they also move out that way? So again, turning to the experimental evidence, here are images from a seminal paper from 2012 from Science Translational Medicine by E. et al. And what they observed, so what you're looking at the top row of the images, you're looking at the brain surface, and in a brain, an arteriole that penetrates into the brain. And this is the lower plots are at later time on the veins on the venous side. So let's look at the top ones first. So what they found was that this green tracer at first appeared in perivascular space on the surface, and later as these penetrating arterioles around 10 to 30 minutes after the injection. And then hours later, they found tracers on the venous side in the veins inside the brain, and also on the outside, on the brain surface. And so this led to what's now known as the glymphatic theory, or the glymphatic hypothesis, which states that really the lymphatic system of the brain is coined the glymphatic system, and it's a system in which tracer enters the brain via perivascular spaces on the brain surface and then into the brain. It travels through the tissue and then exits the brain via perivascular spaces on the venous side. So this is a theory that's gained a lot of interest, but I would say that there's very many open questions about it. And in particular, if you look at these images here, and the interpretation of these images as a tracer enters the brain on the arterial side, travels through the brain tissue and exits at the venous side, that's also something one maybe could make other observations related to. Okay, so we wanted to try to have a look at whether there were other characteristics of peri-arterial and perivenous spaces that could lead to such a delay in observed tracer. And what we wanted to look at was the effect of geometry. So if you now look at the images here in A and B, these are images of human perivascular spaces from our collaborator Eric Backer and his colleagues. And A shows an artery, the circle. The dark green surrounding is a cross-section of the perivascular space surrounding it, and then you have the brain below here. While B shows the similar thing on the venous side. So this more oval ellipsoid here is a vein, and then again you have the darker perivascular space surrounding it. And we now ask the question, well, let's take these two geometries, assuming these are representative for the arterial and the venous side, and impose our favorite 1.46 millimeters of mercury per meter gradient in the axial direction. What type of flow would that induce? Okay, so that's what we did. We considered these two image-based geometries, A1 and V1 for the perivascular space surrounding the artery and vein, and A2, V2 for idealized versions of the same. And then we looked at the velocity in a line crossing through these geometries, and that's what's shown in C here. So essentially, there's the velocity over a line through these geometries. And what we see in the A1 comparing to the V1 geometry is that really you do, of course, get velocities many orders of magnitude higher in these arterial geometry compared to the venous geometry, just by imposing the same axial pressure gradient. And what consequences would that change in velocity have for transport? Well, again, if we now think of this channel, we extend it to a 5 millimeter long channel, which is what they measured in the original experiment. And you simulate injection at the beginning of one of the channel, and then you observe how it distributes in this channel under diffusion by its diffusion coefficient of the tracer injected and by the CSF flow that we just computed. And we can look at, so here's the, this plot shows the concentration at the measurement size. So at the end of the channel over time. And if we again look at A1, the first curve here, we can look at that, well, it'll hit at 70% concentration about maybe 15 minutes. While for the venous geometry, it would hit 70% maybe after 90 to 100 minutes. So it would take much longer to travel through this perivenous space. And if you recall, this delay is very comparable to what they originally observed experimentally. And the later appearance of tracer on the perivenous side could perhaps be explained by geometrical differences, by the characteristic sizes and shapes involved, rather than that the brain has traveled through, that the tracer has traveled through the brain on the way. So in any case, one thing is very clear, and that's what's happened on the venous side in terms of spaces and transport of pressure is very under investigated. A lot more attention is needed there. And I think it's hard for us to say much more about this without more experimental evidence on the venous side. Now, let me sidestep for a moment. I don't know if any of you remember the movie The Hunt for Red October. This is a favorite movie of mine, a stellar movie. It made a very strong impression on the teenage me. And in particular, it ends with this beautiful quote. And the sea will bring each man new hope, as sleep brings dreams. The movie attributed this to Christopher Columbus, but as far as I've been able to tell, and meaning I asked ChatGPT a few years ago, that claim is considered rather dubious. So I've attributed it to unknown here. Well, why do I bring this up? Well, why are we so interested in this solid transport in the brain, really? And one reason we're interested in this is its relation to why we sleep. So why do we sleep, really? Well, one hypothesis is that we sleep in order for the brain to have a chance to clean up. And this hypothesis hit the news with a bang in 2013 via a science paper by, again, this group from Mike and Nedegard. It's a paper by Xi et al. From 2013. And this idea was again launched based on this type of tracer experiments that we've been looking at up till now. And in particular, in one of these experiments, they observed that if you have this tracer injected into wakeful animals or sleeping animals, meaning mice, the tracer moved much, much, much faster into the brain in the sleeping mice than in the awake mice. So that's what's illustrated here, is that the green illustrates how far the tracer moves for the sleeping ones, while the orange and yellow illustrates how far it moves in the same amount of time for wakeful animals. And they also found that the tracer was cleared from the brain about twice as fast in sleeping mice. So up till now, we've really been looking at flow and transport of the surface of the brain mainly, and a little bit into the brain, but mainly on the surface of the brain. And the surface brain provides an intriguing interface between the brain itself and this CSF-filled surrounding that's in direct communication all through the brain and also down into the spinal canal. And if you recall that the tracer in most of these experimental or clinical studies, it's not injected within the brain itself. It's either injected in the spinal compartment, way down in the spinal cord, or for the mouse, it's injected in the cisterna magna, which is also not inside the brain itself. So it's actually not that obvious if these changes that's observed during sleep are in fact due to changes in transport parameters of the brain itself or of the surrounding cerebrospinal fluid. And this point was made in a study by Ma et al. From the group of Stephen Kroll. So again, they studied tracer transport with the tracer injected in the CSF, and they observed how much tracer that reached the brain surface itself and the perivascular spaces on the brain surface. That's the top row of images here. And they observed how much that reached the brain tissue itself. And they made two very interesting observations. They considered awake animals, and then they're considered two different anesthesias as proxies for sleeping mice. So these are anesthetized mice, kind of sleep proxy mice. And the observations they made was that first, the spread of tracer to the perivascular space on the brain surface was proportional to the amount remaining in the CSF of the surroundings. Not too kind of controversial in a way, but important to note. And they also observed that this amount was inversely proportional to how much that had entered into the systemic blood. So how much had kind of left the brain and the cranial system. Okay. And the reason why this is interesting, because if you combine these 2 points, is that an alternative concept to sleep enhances transport inside the brain. You could be that in the sleeping mice, there is less tracer that leaves the CSF surroundings, and therefore there's more tracer available for moving into the brain. Now, this is also something that we looked at computationally as part of a bigger study led by Martin Hoodkjøp, a PhD student, soon to wrap up his PhD in our group. And he considered a patient-specific computational model of cerebrospinal fluid flow in the subarachnoid space and ventricles surrounding a human brain. And in his model, he considered the flow of CSF just driven by the production of CSF. CSF is believed to be produced deep inside the brain and traveling through the ventricular system and out into the subarachnoid space. And so he simulated the flow of CSF driven by a production of CSF in part of the domain and several different possible efflux routes. So the one I'm showing here, I think there was an efflux in the front here, essentially, associated with a nasal cavity. And then we coupled these flow simulations with some diffusion convection simulations of solute transport, with the solute injected in the lower intracranial space here, kind of corresponding to an ejection in the spinal canal that's then moved up. And what you're looking at here are sample simulated concentrations after 6 and after 24 hours after the baseline, with this one choice of plausible efflux pathways. So let's consider this the baseline. And let me tell you that it's known that CSF production is decreased during sleep, or vice versa, it's increased during wakefulness. So we can ask, what pattern will we observe if we increase the CSF production rate? Well, let's double it. So that would correspond to an increase during wakefulness. So here you have doubled CSF production. So you can think of the upper ones as sleep and the lower ones as wakeful. And then you observe that this increased CSF production gives reduced availability of CSF on the surface, and as a constant, reduced trace influx during wakefulness compared to a sleeping configuration. And here you can see, this is an illustration of how you can see what seems to be a very reduced influx during wakefulness, which really doesn't have anything to do with changes in the properties inside the brain, but the changes in the CSF compartment surrounding it, which highlights the importance of really also looking at the fluid compartment surrounding the brain. Okay, so with that, let's move into the brain itself. So perhaps what I've shown previous doesn't tell the whole story either. So let's really get to the coin of the matter in a way. What are the modes of transport within the brain itself? And let me go back to some of the images I showed you at the very beginning again, with a bit more detail. These are images courtesy of Christian Eide and Geir Ringstad, also University Hospital. And they are images showing tracer signal intensity here in one human subject. And the two rows are from different angles, as before. Blue is low, red is high. And clearly you see that the tracer seems to spread into the entire brain from the surface and inwards. And you can also see that we're dealing with noisy data that are relatively high resolution in space, but rather sparse in time. So these are the 4 time points we have in 24 hours, and we also have one at 48 hours and one at 72 hours. So relatively sparse in time. So our task as computational mathematicians are, what can we deduce about transport parameters within the brain itself from these images? And in order to address that, we, meaning again, 2 very talented PhD students, Vegard Vinje and Bastian Sapp, created these patient-specific models of tracer spread in the brain. So from these, we have around 20 patients, and we have images of their brains at baseline, which we use to create meshes conforming to the outer surface and the inner surfaces of the brain. And the baseline hypothesis, knowing about the blood-brain barrier and the lack of lymphatics within the brain, is that solute spreads by diffusion within the tissue. So we're interested in tracking and simulating this concentration C, such that the classical parabolic diffusion equation holds with the diffusion tensor D in the brain omega, where all positive times T. And to compare the simulated and the modeled, simulated and experimentally observed tracer concentration, what we do is that we map these signal intensities from the image data onto concentrations. We linearly interpolate them in time, and now prescribe these as just boundary conditions for our parabolic equation. And then we're interested in observing how the concentration evolves inside the brain. Now, what about this diffusion tensor D? Well, the bad news is that brain tissue is heterogeneous, it's isotropic, and it's highly subject specific. The good news is that measuring the diffusion tensor in brains is a relatively standard kind of imaging module. So we actually have also DTI images and DTI data for each of the patients that we use to inform this diffusion tensity. Okay, so what do we find? Well, in order to quantify what we find, we look at the amount of tracer in gray matter, which is the integrated amount of tracer of the outer outer relatively thin layer of the brain, and the total amount of tracer in the white matter, which is the deeper brain regions. And here the data are given as points, and the simulation results are given as curves. So this is the amount of blue is the amount of tracer in the gray matter over time, and green is the amount of tracer in the white matter over time. And let's look at the influx phase first. This is kind of the up till maybe around 20, somewhere between 10 and 20 hours. And what we can see is that clearly the diffusion captures quite a bit of what's going on here. The simulated results do not match the data perfectly, but quite a large portion. But it seems that it underestimates how much tracer that has moved into the tissue in the early phase. And similarly, it seems to underestimate how much the speed of tracer leaving the brain at later times. So if we just stick with this model but dig a little bit deeper, we could ask, what if either our diffusion coefficient is off by some factor, either due to measurement errors or something else? And modeling wise, we could represent this by an effective diffusion factor alpha. So replacing D by some alpha D. And if we try enhanced diffusion, so alpha 1, 2, 3 maybe, we observe that we're able to match the influx phase almost perfectly, but then not the outflux, the clearance phase. And we'll see similar things. You'd use a lower alpha. You can match clearance, but not influx. So our conclusion was that with diffusion alone, we were not able to match the clinical observations in a satisfactory manner. Okay, so now the plot thickens, and this is where it starts to get mathematically interesting. What about other mechanisms like convection? Is there a convective flow in the brain tissue itself? Well, 40 years ago, Helen Serre and her collaborators did indeed find a bulk flow of tissue fluid, so a convective flow within the brain on the order of 5 to 15 micrometers per minute. So that was 40 years ago, but say 5, 10 years ago, others, like the team of Berkman at UCSF, found no evidence of any convective flow whatsoever in the brain. And as you can tell, this has been a point of controversy with discussion raging back and forth. And I think that one reason for the controversy is really that both experimentalists and clinical work use these tracer experiments as a proxy for understanding the tissue fluid flow inside the brain. But in fact, this setting also defines an interesting mathematical and computational question, which is, given this set of MR data, given this set of tracer concentrations at some time points, the structural data, the diffusion data, can we identify and quantify this convective velocity field phi? Okay, so let's jump into this problem setting from a technology perspective. And detecting movement between pairs of images is really a classical problem setting in computer vision. It's known as the optical flow problem or determining optical flow. And the concept is that given images C1 and C2, or more precisely scalar fields C1 and C2 at different times T1 and T2, we're interested in finding a vector field phi that maps C1 to C2 in some sense and in some optical sense. And we express this as an optimization problem. So we're interested in finding a suitably smooth velocity field phi that minimizes an objective functional that's given as the sum of two terms. One is a regularization term that says something about how smooth this vector field should be. So for instance, an H10 seminorm here, or an H10 norm, I would say. And then you have one term measuring how well phi and the concentration C matched whatever transport relation you're interested in mapping to. And here, in the classical optical flow case, one considers a kind of a pure transport equation. And then you can solve this optimization problem of finding an optimal phi that minimizes this functional via, for instance, the Euler-Lagrange equation. And one version of this technique, not with the H10 seminorm, but with a regularization term that maps that mimics the kinetic energy, is a classical, very classical problem called the Monch-Kantorovich mass transfer problem. And it was brought back and studied in a pivotal paper by Benamou and Bernier in 2000. And exactly this technique with exactly that kinetic energy regularization has been used, in fact, in this setting of mapping kind of lymphatic flow fields by Tannenbaum and Benavist for analyzing tracer transport with high temporal resolution data in mice. But for us, we found really two problems with the formulation as written here. And the first was that the method as is does not account for diffusion. And clearly we've seen that diffusion does play a substantial role. And the second is that all of our numerical experiments were very sensitive to the choice of regularization parameter. We were unable to obtain any meaningful results that were robust in any regularization parameter regimes. So what we instead did was to consider an alternative formulation that we call an optimal convection diffusion, OCD approach, where we still have the same setting. We're giving two scalar fields images C1 and C2 at times T1 and T2. And now we consider a slightly different optimization problem. Now we want C and phi to be related such that this diffusion convection equation holds. But at the same time, we assume that we start with the data given at T1 as the perfect data, but we want to match the data at T2 approximately. We tried various approaches for solving this problem. What we ended up doing was most robust was this iterative optimization approach, where we considered a reduced functional and solved this iteratively, starting with phi equal to 0 and then incrementally updating phi, for instance, using something like BFDS. And this worked very well for us. It was robust, gave solutions for all patients, and robust with respect to choice of regularization parameter. Well, what did we find? Well, as I mentioned, here's one example of the reduction in the objective functional as a function of the number of iterations. We see we were able to match the data well. And I wouldn't trust my estimates for the L infinity norm of the velocity, but we can compute an average speed reasonably well. And you can see here on your right, this resulting one of the velocity fields indicating what are the preferential velocity pathways in the brain. Okay, so now some of you may be thinking, this is very interesting at all, nice graphics, but weren't we really talking about the hunt for red October and why we sleep? Well, here's the twist to the story. Remember, we have these patients been rolled in and out of the MR machine. And in fact, there's two patient cohorts. There's 11 or 12 patients that have been at the hospital, rolled in and out of the MR machine, and then been allowed to sleep happily during the night. And then there's one group of around 8 patients who did the same thing with the MR machine, but were not allowed to sleep on their first night in the hospital. So they were sleep deprived. And for these two patient groups, the tracer cleared differently. In the sleep deprived cohort, the tracer lingered longer in the brain, and the tracer cleared slower by about a factor of 2. Actually, just as for the mice in the original Xi et al. Study. Now, the really attentive of you may ask, well, was this because the tracer lingered longer on the surface of the brain in the sleeping humans? And is it really due to differences on the availability on the surface? And of course, we checked that. And no, it seems that it was not. We did not find any significant difference in the amount of trace from the brain surface between the two groups. So can we instead estimate a difference in tracer mechanisms within the brain? Well, if we just use this technique I just described to examine the convection velocity for all of these patients from 24 to 48 hours, 2 images, 24, 48 hours, estimate the convection velocity and compute the average velocity magnitude. If you then look at the darker blue here, tells you something about for each of the patients, the variability has to do with the choice of regularization. The light blue and dark blue are high and low rest meshes. What we can see in the ballpark we're talking about is that we seem to be getting average velocities on the order of 1 to 8 micrometers per minute. And the interesting thing is that this estimation really captures also the reduced flow speed. In all relevant regions of the brain, we found that the sleep-deprived patients had reduced convective velocities, and it was reduced by about a factor of half. So that's next natural question is, where does that flow on the order of some micrometers per minute come from? And I'll leave that as a cliffhanger. I will not go into that. We have some recent work on interacting brain waves, coupling chemical, electrical, and mechanical effects that at least gives a partial answer. Okay, so wrapping up, I've presented some of the research we do in my team here at Simula relating to solid transport, cerebrospinal fluid flow optimization. We look at many more aspects as well. I'd be very happy to discuss any of these with any of you. And if you realize that what you really wanted to do in your research career is to work with computational modeling of the human brain, we have written a very accessible introductory reference called Mathematical Modeling of the Human Brain from Magnetic Resonance Images to find it on the simulation. That's open access and freely available. And we're also writing a follow-up volume on this manuscript. So let me end there. Thank you for your attention, and give some additional credit, especially to all of my ongoing or previous PhD students, Maurice Kassmann, Ada Ellingsrud, Martin Hornkjøl, Lars Magnus Rognes, Vegard Vinje, and Bastian Self. And thank you so much to the European Mathematical Society and Professor Aranjo for the beautiful cover illustrations. Thank you.